Good afternoon, everyone, and welcome to another session of Food for Sustainability webinar cycles. And during this cycle, we are trying to understand whether we can really reduce greenhouse gas emissions in ruminants. Probably you know me, I am Claudia Costa, the coordinator of Food for Sustainability Academy, and I will be moderating the session today. I hope you enjoy. We have three very interesting speakers, and we'll be addressing the topic of natural sources of feed for low emissions in ruminants. And to address this interesting topic, I am joined by three experts, which I truly thank for their availability to join our discussion. And with us, we have Alejandro Belanche. He's a researcher at the University of Zaragoza and an expert on molecular techniques to describe modes of action in nutrient intervention in ruminants. Thank you, Alejandro, for being here. We also have Pekka Huhtanen, who is a senior advisor at the Natural Research Institute in Finland, the Luke Institute, with an expertise, among other things, in forage evaluation, feed intake, and cell wall digestion. Thank you, Pekka, for participating. And from Portugal, we have Henrique Ramos. He's a consultant for the blue economy, specializing his work on the super algae, Asparagopsis taxiformis. But he's also a very experienced sailor and lives in a beautiful group of islands in the middle of the Atlantic, which is the Azores, which I invite you all to visit if you don't know Azores. And Azores has this very particular characteristic of having more cows than inhabitants. So many thanks to you all for being here. And for those that probably don't know yet, let me just give you a couple of words about Food for Sustainability Academy. So the Academy is the learning hub of Food for Sustainability, dedicated to knowledge dissemination and awareness raising on the topics that contribute to Food for Sustainability mission, which is the sustainable intensification of food systems towards more health and well-being. And naturally, we cover topics such as circular economy, sustainable farming, ecosystem services, and functional nutrition. And if you want to know more about our projects, you can always go to our website, or eventually, if you prefer, just sign up to our newsletter. We have here our communications team. Also, Cecilia Franco will be here to assist you in this process if you wish to join. Also, I would like to start by thanking all Food for Sustainability team for their commitment and support towards these webinars. Also, words of thanks to BGI, Building Global Innovators, for the dissemination assistance, and I especially name BGI CEO Gonçalo Amorim and BGI's marketing manager Eduarda Monteiro for their continuous support. Of course, I could not go without acknowledging the work of our multitask byproduct valorization team, who relentless have worked to bring together these sessions to you. And I name Rita Silva, our microbiologist. Rita, wave at us. Hello, Rita. Silvia Moreira, our food science PhD. Hello, Silvia. And Daniela Fonseca, our sustainable chemistry PhD. Hello, Daniela. I can also see on the screen Margarida Palma, who has been very supportive. She's our PhD in microbiology. Thank you so much to you all. Before we proceed, let me remind you that some of the house rules to run these webinars. So the sessions are recorded. So if you do not wish to appear, please just remain with your cameras off. We have several demands for later viewing of these webinars, so we keep these sessions recorded. Also, questions are very welcomed, and you know that these sessions are more rich and we can all gain from each other's knowledges and by sharing the questions. So questions are encouraged. Just for time management, we need to have them in writing. We will use Slido for this. When we go to Slido, you will have a dedicated tab just for your questions, and I will pick your questions and address them to the speakers. If you have troubles in writing the questions in Slido, please use the chat here in Teams, and someone from the team will place the questions in Slido. Okay? So thanks for taking part. And do we all know Slido? Slido is very easy to use. We just need to type in your tab www.slido.com. And once you have this window open, you will find a hashtag box, and you just need to insert, as Cecilia put up here in the screen, F4S Academy, and you should be easily in Slido. So Slido, it's great. It will assist us with the questions, and will also assist us with a small quiz that the team has prepared for us to test our knowledge on the topics that we are going to discuss. So this is again a moment. This is again a moment to an opportunity to start learning from the start. So, and our first question relates to dairy cows and the energy involved in milk and meat production from these animals. So, producing these elite dairy cows, the super cows for milk and meat production, is in terms of energy equivalent to how much more in terms of the maintenance requirements from the animal. So producing milk and meat is how many times more demanding in terms of maintenance for the bovines. I think this is a tough question. Let's see if our participants know, but I will tell you that Pekka has spent a lifelong research looking into animal efficiency, namely on all energy sinks, and I think you will be pleased to let you know a little bit more about all the different pathways to growth, milk production, digestibility, and to metabolize methane. That, of course, requires energy, and that, of course, needs to come from feed. And I just, while you're still voting, let me add that I think this is a very important relationship, because it's point us already to the potential to understand how a higher milk production is indeed related to feed efficiency. And what I can see from the screen is that we have a very well-informed audience. It's 4 times greater. Congratulations. I'm really impressed. So this is going to be a promising webinar. Thank you for this. So let's see if we are that knowledge about the second question. Thank you. So our second question relates to natural ingredients. And the question is, what are some of the plant metabolites that can inhibit methane production in ruminants? Saponins, essential oils, flavonoids, or flavonoids, proteins, or essential oils, or terpenoids, essential oils, or polysaccharides. What is your guess in terms of these metabolites? And Alejandro has specialized in understanding how the different mechanisms can be triggered to reduce rumen methanogenesis. And I think he will let us know a little bit more about how antimicrobial and nutritional properties have an impact, and also the methanogene community are all impacted by these plant metabolites. And do we have all the questions? So we are talking about saponins, essential oils, and flavonoids. And you're absolutely right. Again, a very knowledge audience. I think we need to increase the level of difficulty of these questions, or congratulate our audiences, because definitely you're spot on. This is the right response. This is the secondary metabolites that actually contribute to inhibiting methane production. So let's test our audience once more. And our last question relates to our super famous seaweed, the Asparagopsis taxiformis. And Keeling and colleagues, they had made a strong claim about methane emission reductions in beef cattle by affecting rumen digestibility. Do you have an idea of what the number is? And this is the topic that Henrique has been focusing his work. So he's been working on seaweeds, and he's been working at the most efficient way to feed animals with Asparagopsis. And I would dare to say that this figure is something that's encouraged his daily work. And although we have other alternatives, commercial alternatives for this essential oil, this is a very promising number. So do you have an idea of what the number is? Okay, so we've learned something. So according to Kinley and his colleagues, 2020, this essential oil can actually reduce the digested retained methane emissions by 98%, which is a very impressive figure, but it's something that is definitely very promising and encouraging. And maybe, Henrique, you can explore a little bit more about these figures. So we have tested our knowledge. I think everyone passed this very short quiz. Thank you for the team. And so let me now turn to our speakers and let me present them in a little bit more detail. So the first speaker is going to be Pekka Huhtanen. And Pekka received his PhD from the University of Helsinki in 1984. Apologies, 1987. And from 87 to 94, he worked as junior and senior scientist at the Academy of Finland, which also included a postdoc at the US Dairy Forage Center. From 94 to 2007, Pekka was a professor and research professor at Agriculture Research Center. He was also an associate professor at Cornell University between 2008 and 2009, and a professor at the Swedish University from 2009 to 2022. He's currently a visiting professor at the Natural Research Institute in Finland. In 2003, Pekka was granted Doctor Honoris Causa from Estonian University of Life Sciences. Pekka studies have focused on different aspects of ruminant nutrition, including forage evaluation, feed intake, cell wall digestion, nitrous metabolism, and production responses. Recent years, the main emphasis has been in methane and feed efficiency, and this is what Pekka is going to talk about. And during his career, he published about 230 papers in peer-reviewed journals with an H index, according to Google Scholars, of 71. Pekka, thank you so much for joining us. And our second speaker is Alejandro Belanche. He's a veterinarian, which conducted his PhD at the University of Zaragoza, Spain. He has 8 years of international research experience at the Institute of Biological, Environmental and Rural Science in the UK, and 5 years at Estación Experimental de Zaiden, which is obviously in Spain, Granada. And from 2021, Alejandro is conducting his research and also teaching at the University of Zaragoza. His research career is devoted to the optimization of rumen function through nutrition in order to improve efficiency and sustainability, and to decrease the environmental impact of livestock agriculture. He has been working with the implementation of several state-of-the-art molecular techniques that allow to describe the mode of action of the nutritional interventions. Over his career, Alexander has been involved in 29 research projects and 50 research contracts in the livestock sector. He has published 57 peer-reviewed papers, 17 popular science articles, and 13 book chapters. As a result, Alexander has an H index of 24 and appears in the last Standards for ranking as one of the most prominent researchers in his research area. Currently, Alejandro is the editor in Frontiers in Microbiology, co-leader of a working group of the Global Research Alliance flagship project on feed additives, and a member of Remedia Executive Board. And Alejandro is going to present how use of plant secondary metabolites can be used to decrease rumen methanogenesis. Thank you so much, Alejandro, for being here with us. And our third speaker is Henrique Ramos. He is graduated with a passion for geography and the ocean, and has brought together all this experience in seaExpert, where he's dedicated to providing services and consulting for the blue economy, as well as marketing algae for cosmetics, animal feed, and research industries. More recently, seaExpert has focused part of its work on the super algae Asparagopsis taxiformis, mainly for its anti-methanogenic effects, but also for this cosmetics application. Currently, and within the scope of sustainability, Atlantic Biotech is also involved in the Mar for Terra project to transform algae and waste from the agro-food industry into biofertilizers and soil conditioners. Henrique, it's a pleasure to have you here. Thank you so much. And I think enough of me. We have a very promising webinar ahead. Pekka, I'll now hand the floor to you, and I invite you to share your screen and start presenting. Thank you all.
Now you can hear also.
Yes, perfect.
Okay.
Thank you.
Okay, first, thank you for invitation for this webinar, and good afternoon to everybody. I will talk about feed efficiency and methane, how they are connected on each other, and a little bit discuss, can we improve feed efficiency by decreasing methane production, or vice versa, that can we reduce methane emissions, especially per unit of product, if we can improve feed efficiency. Because we all know that methane is a strong greenhouse gas, and its global heating or warming potential is about 28 times greater than for carbon dioxide in this 100-year period.
Pekka, sorry to interrupt. We are not seeing your slides. I don't know if you attempted to move your slides. We're still in the first slide.
Okay, what it is? What happened?
They were moving. Pekka, if you find it hard,
okay.
Yes, perfect.
Yep.
Great. Working.
Yeah, the contribution of enteric methane production to total anthropogenic or human-made methane emissions is estimated to be 27%, but the values are a little bit variable in the literature, but probably this represents average value. Also, there has been intensive research for, let's say, about last 20 years aiming to reduce methane emissions to understand better how methane is produced in the rumen. We can have different strategies how to decrease methane production. We can divide them into short-term strategies and long-term strategies. Of course, very efficient short-term strategies that we simply reduce number of ruminants in the world. That will certainly decrease methane emissions from enteric fermentation. Of course, it has been a lot of research, diet manipulation, changing diet composition. Probably the main focus has been fat supplementation or feeding more concentrate supplements. But I think in this area we should look the whole picture, whole greenhouse gas emission, not simply enteric methane, because like for example producing fat that is usually replacing concentrate in the diet, its greenhouse gas emission can be much greater than for grain production, and also for concentrates. It decreases methane emissions, not so much with typical dairy cow diets when we are not feeding extremely high concentrate diets. But if we take into account soil carbon sequestration, it can actually be that forage-based diets are more efficient in terms of greenhouse gas emissions than concentrate-based diets. And of course, there is very intensive research looking different kind of feed additives to decrease methane production. So far, there is not very many practical applications that are used in a larger extent. And then, of course, we can have long-term strategies. There have been in interest some countries breeding low-emitting animals. In New Zealand, main emphasis have been breeding low-emitting sheep, but there are also research projects in Europe trying to decrease methane emissions by animal breeding in dairy cows. And of course, other long-term strategies to improve production efficiency that can contribute to improved nutrition, breeding for higher production, improved management, that all increase production level, and then producing same amount of animal products from smaller number of animals. And that is actually what has happened in most Western countries during the last decades. Of course, it is if we could decrease methane emissions in ruminants, of course, then the total atmospheric heating potential would decrease. If we look the numbers there, that this methane production and methane sinks, they are quite close to balance. So if we could decrease methane emissions from, for example, from ruminant production, then it could lead to situation that methane emissions are smaller than methane sinks. Of course, there is some variation in the numbers, but during the last years anyway, the atmospheric methane concentration has been increasing. But on the other hand, world ruminant population has been quite stable. That we probably can't blame ruminants for this increase in atmospheric methane concentration. We don't often remember that the methane is methane from ruminants. It is biogenic methane. It is recyclable. It takes about 10-12 years when it is converted to carbon dioxide, and then carbon dioxide is used in photosynthesis by plants, for example, growing grass. Of course, plants don't take directly methane. It is first converted to carbon dioxide. And then, of course, these animals are eating grass and producing methane. So if the situation, if the system is in steady state, then the atmospheric methane concentration does not decrease. But of course, if we can decrease this part here, this process here, then it will lead reduce amount of methane from enteric fermentation. But it is different from if you burn fossil fuels, that the turnover time of carbon dioxide is estimated to be about 1,000 years. Yeah, if we look this how feed efficiency can influence methane production, of course it depends how we express methane. If we look total methane or per unit of product or per unit of feed intake. But I think that if we want to produce certain amount of animal milk or beef, then we should look this efficiency or what is called methane intensity, that is amount of methane produced per unit of product. In animals, feed efficiency is expressed different ways. Typically, this feed conversion efficiency, that is energy corrected milk divided by dry matter intake. Of course, this has certain limitations, especially if the animals are mobilizing body fat, then we may have high feed efficiency, but it is because the animals are using body tissues. And it is mainly affected by dilution of maintenance requirement to larger volume of milk. More recently, feed efficiency has expressed as residual feed intake. That is a difference between observed feed intake and feed intake predicted from energy corrected milk yield in dairy cows, or body weight gain in growing animals, metabolic body weight that represents maintenance requirement, and body weight change. And of course here, if the animals consume, if the value is negative, then it means that animal is efficient, and vice versa, positive values mean low efficiency. We can also use residual energy corrected milk yield, that is some kind of opposite term to residual feed intake. It is observed production minus predicted production, and production is predicted on feed intake, metabolic body weight, and body weight changed. I think from farmers' point of view, this is probably easier to understand because positive values mean a good thing and negative values mean bad thing. We made a modeling exercise to look this development of methane production in Finland. That we have good statistics of number of animals, total production, and also in cow controlling system we can get quite good picture how the animals have been fed at the different time periods. This just or in this figure we have total number of cows that was highest in about 1965, then it was clearly about 1 million. There it has decreased to about 250,000. So at the same time, total milk production per cow has increased from about 3,000 to about 9,000 kilogram. In control herds, it is little more than 10,000 at the moment. But there has also been some decrease in total milk production. That here we had some overproduction. Now it is quite close to self-sufficiency. I think that this type of development has taken place in probably most Western European countries. Then if we look at what has happened to methane production, total methane production was peaking in 1965 when we had the highest number of cows, and then it was about 110,000 tons, and it has decreased so that it is nowadays about 37% what it was 60 years ago. But of course per cow this has increased both when we take without, or if we just look dairy cows, or if we take replacement into account. And of course this is because the cows are eating much more nowadays than what they were eating before. Then if we express methane production per unit of milk, both taking into account replacement or without replacement and also separately replacement. And there it has also been very positive trends. It is now about 37, so almost 40% lower than it was 60 years ago. At some periods there was a little bit negative development in replacement because the cows had less production years, but now it has also been positive trend here, that so the lifetime production has increased, both because of production has increased per cow per year, and also the longevity of the cows has improved. So if we just look this how much the cows were producing milk per 1 kilogram of dry matter intake, and how much methane they were producing per kilogram dry matter intake, taking also replacement into account. And if we make XY plot of these two, we can see that there is very strong relationship. So that if we can improve production, it doesn't matter if it is nutrition, breeding or management, it will lead to decreased methane emissions per unit of product. This so far, this animal breeding or breeding dairy cows, it has mainly been breeding for higher milk yield without no specific attention on true biological efficiency. So it has mainly been dilution of maintenance requirement to create a volume of milk with increased milk production. So it is improved genetics, improved nutrition and improved management. But actually there has not been any positive development in biological processes, digestion and metabolism. And actually maintenance requirement per unit of metabolic weight, it seems that it has increased with the increased production level. We can probably say that the cows have bigger engine, these high producing cows, that the intestinal and liver tissues, they comprise a greater proportion of body weight of the cows. Digestibility has slightly decreased because the animals are fed higher level of intake, but that has partly been compensated by reduced methane production per unit of intake with increased intake. We have seen that there is considerable between cow variation in feed efficiency. We made meta-analysis of large respiration chamber data. Our PhD student, it had about 840 cow period observations. When we ranked the cows according to residual feed intake, then the best 1/3 of the cows, they consume 2 kilograms less dry matter than the poorest cows when they were ranked according feed intake, what means that they were producing same amount of milk, same metabolic body weight, and they had same energy balance. And in respiration chamber study, it was really measured energy balance and not estimated from body weight change. And if we look the residual energy corrected milk yield, this probably demonstrated even better, that at same dry matter intake, same body weight and energy balance, the most efficient cows produce 6 kilograms more energy corrected milk than the lowest efficiency cows. So this reflects the same in figures. Here is dry matter intake. When the cows were ranked according to residual feed intake, then of course low efficiency cows, they were eating a little bit more than 2 kilograms more dry matter than the highest efficiency cows. And when they were ranked according to residual energy corrected milk yield, by definition, they were then eating the same amount of dry matter. When we looked milk production or energy corrected milk production, then when they were ranked according to residual feed intake, there was no difference in production, but they were eating less. And when they were ranked according to residual energy corrected milk yield, they were eating same amount, but they were producing about 6 kilograms more energy corrected milk. What explains these differences between cows in feed efficiency? Roughly about 1/3 of this improvement was in converting dietary gross energy to metabolizable energy, and that was mainly because of improved digestibility. In methane, there was minimal differences between the cows, and also urinary energy. So this main difference in metabolizable energy, it was because of about almost 3% unit higher diet digestibility. But the main difference was in heat production, both ways when the cows were ranked according to residual feed intake or residual energy corrected milk yield. High efficient cows, they produced more heat. And then especially if we look this residual energy corrected milk yield, high efficient cows produced more milk, and then they produced less heat from the same amount of feed. So they were more efficient, but we don't know exactly if they had smaller maintenance requirement or if they were using metabolizable energy more efficiently for milk production. But it is more like an academic question. Then if we look methane intensity, this how much methane was produced per kilogram energy corrected milk yield. The differences were smaller when we expressed differences in efficiency by residual feed intake, but it was still quite big difference. But especially if we look residual energy corrected milk yield, so that these cows were eating same amount of milk, producing more milk, and then they produce about same amount of methane, but when they produce more milk, then per kilogram of product, they were producing about 20-25% less methane. So what are, if higher feed efficiency has so positive effect for methane emissions, why the cows have not been specifically been bred for improved feed efficiency? The simple explanation for this is that measuring feed intake in practical farms is very laborious, and it will be very expensive. And if we have just short period of measurements, the data may not be accurate enough to detect these differences, and it also needs feed analysis. There are recently some alternative systems that are developed for this, especially Danish and Nordic breeding companies. They are looking for this 3-dimensional camera to estimate feed intake. And then it is this green feed system that was originally developed for measuring methane emissions, but also the first versions, they were measuring also carbon dioxide production, and the latest version, they also measure oxygen production. It is actually same system as respiration chamber, but it is like spot sampling system that cows visit there few times a day, probably from 3 to 5 times a day, probably about 5 minutes. They get a bait of concentrate that attracts them to eat the concentrate. There is constant airflow here in the system, air sampling here, and then the gas is analyzed from methane, carbon dioxide, and oxygen concentrations. And from oxygen and oxygen consumption and carbon dioxide, we can estimate heat production. And it actually looks quite promising. We made here in Jokioinen with 32 cows. We compared green feed and respiration chamber in measuring oxygen consumption and carbon dioxide production. Sorry, I have got both 2 times oxygen consumption figure here, but they are almost like mirror images that correlation was a little bit weaker for carbon dioxide than methane, but I think overall it looks very good, and we can predict from carbon dioxide consumption and milk yield, we can predict dry matter intake quite well. So we made from this respiration sample data, we evaluated this residual carbon dioxide, that is same kind of parameter as residual feed intake, how it correlates to residual feed intake or residual energy corrected milk yield. And as you see that it looks quite promising, that it seems that we can use this residual carbon dioxide or probably residual oxygen consumption or even residual heat production. We can use this as a proxy for feed efficiency. One green feed unit, it can measure about 25 cows at the same time. Then I mentioned earlier that this sounds quite attractive alternative, this breeding for reduced methane emissions, that there are quite large research projects in New Zealand with sheep and Denmark, Holland, with dairy cows, but at least I see some problems in this approach. First, that on farm measurements, that are the measurement methods accurate enough to detect this between cow differences in methane? Because the true between cow variation in methane, when they are eating same amount of dry matter, they are actually quite small. That in methane yield, what is amount of methane produced per kilogram dry matter intake, coefficient of variation, it can be only from 6 to 8%. So it is very small. Some of these on farm systems measuring between cow variation, they have estimated from 15 to up to 30% between cow variation. So this much higher variability means or success that most of this variation is actually random variation related to measurements. Then the people are using this predicted carbon dioxide production as an like a marker, and they predict carbon dioxide production from milk production and metabolic body weight, and this favors inefficient cows and penalize efficient cows. It is because of efficient cows, their predicted carbon dioxide production is higher than actual production, and their true carbon dioxide production is actually below the average, because they use energy more efficiently to milk and they produce less heat and less carbon dioxide. And one concern is that low methane emitters are poor feed digesters, or they digest feed less efficiently than the cows that emit more methane. And this is simply because low emissions are related to short digester retention time in the rumen. And if the retention time is decreased, then of course digestibility will be decreased. I think that it is quite impossible to find cows that digest feed very efficiently and emit little methane, because this digestion rate, it is more feed characteristic than, or microbial characteristic than animal character. Okay, some conclusions. Improved efficiency combined with a reduced number of animals is a sustainable long-term strategy to reduce methane emissions without compromising production. Of course, it can be combined with a specific diet manipulation of feed additives, provided that they don't increase production cost or have negative effects on production. So improved feed efficiency, it can be considered win-win situation. The economy of improved because feeding cost per unit of milk will decrease and emissions decrease. And improved efficiency decrease. Yeah, it decrease methane emissions per unit of product. So to realize all benefits of this, then the number of cows should be decreased. Or if we keep the same number of animals but improve feed efficiency, then we can produce more food from the same number of animals. And certainly improve feed efficiency, decrease methane per unit of product, but reduce methane does not improve feed efficiency. Of course, it also decreases methane per unit of product, but the amount of energy saved from reduced methane, it is quantitatively quite small, and there is very little evidence that this saved methane has improved productivity of animals. And with some additives, there is indication that the feed intake will partly decrease, and so the cows when they get more energy from reduced methane, then they don't need to eat as much as before, and so that feed intake decrease and full benefits in production may not be changed. I think globally improved production efficiency and reduced number of animals is the only realistic large-scale strategy without sacrificing the total food, milk and beef production. Additives can work mainly in intensive Western systems, but they are very difficult to use in grazing systems, for example, or more intensive systems. Okay, thank you very much for your attention.
Thank you so much, Pekka, for such an interesting and comprehensive overview on the state of the art of how we can increase farm efficiency while assisting the planet at the same time, and definitely to bring to our attention, the new metrics that combine both animal feed and methane emissions. And while we wait for the questions for the audience, and I know you've been addressing this throughout your presentation, but these new measurements, how far do you think they are of becoming real tools for farmers, or are we just more on academic level to check for the avenues? And if you may allow me, I would really love to hear just a clarification also for our audience, because you clearly point to the intensive production systems that we cannot achieve much more improvements in efficiency through feed efficiency. So we need different measures. Can you clearly explain this rationale and why we need to look at different measures? Because I think this also is the starting point.
Yeah, you asked about this, how close this is from practice. This, but I think that animal breeders, that they are starting to pay more attention to improving feed efficiency, and I think at least first step for improving this efficiency is that they are paying more attention to body weight of the animals, so that this maintenance requirement is not increasing. But of course, like using this green feed system in practical farms, of course it needs investments and we need to fully see the benefits. But I think if you think about globally, this in our intensive systems, we don't get a huge improvement in methane emissions per unit of product or amount of dry matter consumed per unit of product by increasing production. But if we think about the situation in developing countries where production levels are very low, then even quite small improvement in productivity, they will lead to huge improvements in methane intensity. that it is quite different if we increase milk production from 1 kilogram to 2 kilograms per day. It almost decrease methane intensity to almost 50%. But if we increase from 30 to 31 kilogram, it has much smaller effect.
Okay.
But of course, it is we know this that this would be efficient strategy in developing countries, but animals have many other roles there than producing feed, and so it is difficult to realize in practice.
Yes, thank you. And we have been addressing, the key role that the animals play for the equilibrium of the entire ecosystem. And you correctly pointed in your presentation. We are really running slightly late. Just one question for our audience. I'm going to pick one. What do you think are the future trends in improving feed efficiency? What do you expect to change in the next years?
Yeah, of course, even if we can start selecting specifically for improve feed efficiency, it will be quite slow process. But of course, when we achieve something by animal breeding, it is permanent, and it is like interest to interest when we continue breeding for this. This was one question. What is this between cows? It just means that how different the cows are. That what it is between cow variation. That of course if there is a little variation like in feed efficiency, then there is no point to start breeding it for.
So it has to do with the individual animal. Pekka, I need to stop here. We'll leave these questions for the end if we have more time, because we're running a little late. So thank you so much for your presentation. And now I'll give the floor to Alejandro. I invite to share his screen. Can see your screen? Yes, thank you, Alejandro.
Okay, can you hear me?
Perfect.
Yes.
Okay, so thank you very much for the organizing committee for allowing me to present these results. And today I'm going to talk about the use of plant secondary metabolites to decrease rumen methanogenesis. And first, I want to describe why it's important. And to know that, we need to understand that the ruminants, they have a digestive peculiarities, which is the presence of the rumen, which is a fermentation chamber in which different microbes habit. For example, there are bacteria, methanogens, protozoa, and fungi. Those microbes, they are able to digest fibrous feeds and as well low quality proteins. So they have some advantage in comparison with the monogastric. So when we are feeding ruminants, we are not feeding the animal itself, but we are feeding the microbes. And those microbes are the ones that are going to produce the methane. Of course, during that fermentation, the end products are volatile fatty acid and microbial protein, which represent the main energy and protein sources for the ruminant. So the idea of these nutritional interventions is to take advantage of the peculiarities of the ruminants and to minimize the inconvenience. Among the inconvenience of the rumen fermentation is the inefficiency, because for every 100 grams which are eaten by a cow, only approximately 25% of the nitrogen is transformed into milk. The 75% remaining is excreted as a urine or feces, with the subsequent environmental impact. As well, during the fermentation process is produced methane, which represents between 2 and 12% of the gross energy intake. So decreasing methane, as Pekka just have shown, if we decrease methane, we can as well increase productivity because less energy will be wasted. So here is a description of how the methanogenesis occurs in the rumen. So basically, we have the feed, which is fermented into volatile fatty acids such as acetate, propionate, and butyrate. And during that fermentation is released hydrogen and CO2, both elements are combined by the methanogenic archaea and transformed into methane. So the main strategies to decrease or to minimize methanogenesis can be classified in three. One is to try to decrease the hydrogen production by tweaking, by changing the proportion of the different fatty acids. For example, if we increase the production of propionate, we will decrease hydrogen production, and the opposite will happen with acetate. That's the reason why when we feed concentrate diets, the amount of hydrogen and the amount of methane is lower than the animals when the animals are eating forage. Another strategy is try to redirect the hydrogen towards other alternative hydrogen sinks, for example, through the acetogenesis, through the sulfate or nitrate reduction, or through the fumarate reduction, which ultimately will be transformed into propionate. And finally, the last strategy is to act specifically against the methanogenic archaea, inhibiting the activity of those microbes. And we will see later on the Asparagopsis is the mode of action is inhibiting the archaea. So we have to mention that the methane emissions can be expressed in different ways. For example, we have the methane production, which is grams per day. Okay, that is important to decrease because that is ultimately what is the source of methane. But as well, we have methane yield, which is grams per kilogram of dry matter intake. That is more constant. And finally, we have methane intensity, which is the emissions of methane per unit of product. It could be milk or meat. So some strategies directly decrease methane production. But other strategies, they don't change the methane production. The only thing they do is to increase the productivity. So as a result, we have a dilution of the methane intensity per unit of product. Well, today we are going to focus particularly on the secondary plant metabolites, which is the same than plant extracts or phytochemicals. There are different names for the same thing. And they are plant molecules coming from the plant, which are originally developed as a defense against microbes, insects, and herbivores. But these molecules are able to modulate the gut microbiota and to improve animal physiology. There is a high diversity in structure, bioactivity between the different plant extracts, depending on the type of plant, the part of the plant, the harvesting season, the geographical location, or the extraction procedure. And basically, we can classify these phytochemicals in two. One are the ones which are coming directly when the animal is fed with the whole plant. For example, when the animals are grazing plants rich in tannins. And the other one is if we use the plant extract itself after an extraction procedure, and we add that into the diet. So like this, we can use it in intensive systems. The plant extracts can be classified according to the botanical origin, the chemical composition, the processing method, or even the mechanism of action. And we are not going to talk too much about that because we don't have much time, but I will spend a bit of time describing what is the ideal plant extract. The ideal plant extract should be stable and chemically defined, because some plant extracts they get off very quickly. That is not good. The plant extract, they should be sustainable and economically viable. That means that we need to produce enough quantities of the plant extract in order to feed the ruminants, and as well the extraction procedure should be relatively cheap in order to be profitable for practical use in the livestock agriculture. As well, the plant extract, they should have positive effects in animal physiology, for example, improving palatability, digestion, performance, or decreasing methane emissions. And finally, the ideal plant extract should have persistent and quantifiable effects, because sometimes the effects can disappear along the time. So we will talk about the main categories of plant extract. One of the groups are the essential oils, which are they can be classified in two types, which are the phenylpropanoids, such as the eugenol, cinemaldehyde, and anetol, or the terpenoids, such as thymol, carbachol, or capsaicin. And the mode of action of these essential oils is different depending on which one we are referring to. But in general, they have antioxidant properties. They have as well antimicrobial properties, and some of them they improve the absorption of the nutrients. As well, most of the essential oils they have a pleasant smell, and the oils are extracted by distillation. Most of the essential oils are commercialized as a blend, so a mixture of different essential oils. And the activity is multifactorial against the microbes. They tend to decrease as well ammonia production in the rumen. Here you can see the different essential oils that you can find in the market and what are the effects against the different microbes. And in general, we can say that the essential oils, they have a non-specific antimicrobial effect. Moreover, Casamiglia demonstrate in a review paper that each essential oil they can have different effects depending on the rumen pH, so they are pH dependent. So that's why some essential oils are working fine for a type of diets, but not for other type of diets. So that's why we have to check in our production conditions. In this table, I present a table from a book chapter that we published recently, in which we include the most recent studies in vivo which use essential oils. And we can see that in general, the effect of the essential oils in the dry matter is very small. Sometimes they increase intake because they are highly palatable. And as well, sometimes they decrease methane, but as well, they can increase slightly the milk yield. In a meta-analysis we published recently, we analyzed 23 studies in which dairy cows were fed with agolin ruminant, which is a blend of different essential oils, and we observed that during the first month or so, the animals they have similar milk production, but after 1 month they have approximately 4% higher milk yield, indicating that the essential oils are favoring the efficiency of the production. As well, in some of those studies, we observe a decrease in methane production in about 10%. But we need to have more studies to conclude that this particular essential oil blend is a promising strategy for decreasing methane emissions. Another type of plant extracts are the tannins. They can be classified in two types. One are the hydrolyzable tannins, which are small molecules. They can be obtained from the chestnut, the oak, or the tara. And then we have the condensed tannins. They are polymers, so they are bigger molecules, and they can be obtained from the quebracho, mimosa, or acacia. They have antioxidant properties because they are polyphenols, but the main characteristic is that the tannins bind to the proteins in the diet, and they protect the proteins to be degraded into the rumen. When those complexes arrive to the small intestine, the complex is broken down and the proteins are released. So that's a way to increase the rumen bypass protein. Here, in this review, we observe that the tannins can have a positive or negative effect on dry matter intake because the tannins are very bitter, and if the inclusion rate is too high, the animals will not eat the diet. They decrease, of course, the ammonia concentration in the rumen because the protein is protected, and they tend to increase as well propionate molar proportion. Sometimes they decrease methane, but it's not very consistent, and as well they tend to decrease protozoa concentration. What is more obvious is that the tannins, they change or they shift the nitrogen partition. So the animals fed with tannins, they tend to tend to have a lower urinary nitrogen excretion, and they normally have a higher fecal nitrogen excretion. And that is very relevant because the nitrogen excreted as a urine is more pollutant than the nitrogen excreted as feces, because the urine is evaporated as ammonia and nitrous oxide with the subsequent environmental impact. In a paper, we describe that the use of brown seaweed, different brown seaweed, such as Laminaria digitata or Asparagopsis nodosum, they can have different effects in the rumen microbiome depending on the content of fluorotannins. Actually, Ascophyllum has much higher content of fluorotannins than Laminaria, and as a result, the nitrogen digestion was lower. But we can use that as well to decrease the rumen concentration of certain pathogenic bacteria, such as Escherichia coli. Other type of plant extracts are the saponins. There are two different types as well. There can be steroids glycosides, such as the ones coming from yucca, oats, or asparagus, or they can be triterpene glycosides. For example, the saponins coming from the quillaja, tea, or the alfalfa. The saponins are molecules with high molecular weight, and they have a sugar and a sapogenin, which is the main active compound. The main activity of the saponins is they have anti-protozoal activity. The problem of the saponins is that the effect often is transitory and tends to disappear over time. That is because the rumen bacteria develop the ability to break down the saponin and to inhibit it. So there are different strategies to overcome this problem. For example, intermittent supplementation with saponins, combination of different type of saponins, or saponins chemically modified to prevent this problem. Here you can see what is the mode of action of the saponins. You can see that is multifactorial, but most of the effect is driven by the anti-protozoal activity. And why is important the anti-protozoal? The reason is that the protozoa, they are eukaryotic cells which are living in the rumen, and they have a type of organelle which are called hydrogenosomes. They are similar to the mitochondria, but these organelles produce hydrogen. The hydrogen produced by the protozoa is rapidly used by the methanogenic archaea, which are living inside and surrounding the protozoa. As a result, it has been demonstrated that the concentration of protozoa in the rumen is positively correlated with methane emissions. So if we remove protozoa, we decrease methane emissions. And here we demonstrate that in a meta-analysis about the effect of the defaunation. Defaunation is removing protozoa from the rumen. We observe that the animals without protozoa, they have approximately, on average, 11% lower methane emissions, and as well a higher nitrogen efficiency in terms of microbial protein synthesis. But the problem, if we remove protozoa, is that we can compromise to some extent the digestibility of the fiber, because the protozoa are highly fibrolytic microbes. So we have some advantage and some disadvantages. Here we see the effect of the saponins. In general, we can observe that they tend to increase propionate molar proportion. They sometimes, not always, but sometimes in some studies, they can decrease methane emissions. Of course, the decrease in protozoa is a more consistent effect, and they tend to increase the microbial nitrogen as a result of the elimination of the protozoa, because the protozoa, they intake bacterial protein, so they are competitors with the ruminant for the microbial protein. I want to say as well a few words about the use of probiotics. These are not plant extracts, but as well are nutritional interventions that can be used not to decrease methane emissions, because the use of yeast, for example, or bacteria such as Enterococcus, Bacillus, or Lactobacillus, they don't decrease methane, but they tend to increase slightly the productivity, about 1 to 2% meal yield or meat production. So as a result, we could say that the use of probiotics could decrease to some extent the methane intensity, but not the methane production. Here in this table, I present the probiotics which are allowed to be used in the European Union. Other alternative is the use of lipids. Because it has been described that the lipid supplementation decrease methane emissions. The reason for that is because the lipids have antiprotozoal effects, particularly if they are highly unsaturated lipids. The problem is that we cannot use lipids in a high proportions because as well they have an inhibitory activity of the fibrolytic bacteria. So high supplementation with lipids can compromise the fiber utilization by the ruminant. Another alternative is the use of nitrate, because in the rumen the nitrate is reduced into nitrate, and that acts as a hydrogen sink, is capturing hydrogen from the rumen. And as a result, we can observe a decline in methane emissions. The problem of the nitrate is that it's toxic because it transforms the hemoglobin in metahemoglobin, and if we feed too much nitrate, we can produce toxic effects into the rumen, even the death of the animal. As well, I was going to talk a little bit about the red seaweed, but then we have another speaker that will talk about this. Basically, I can only say that the Asparagopsis is the most promising seaweed, and there are two species, Armata and Taxiformis. And the mode of action of the Asparagopsis is that is an inhibitor of the methanogenic archaea, is the similar activity as the 3NOP, and that is because they have a bromoform. They have bromoform that is inhibiting the last step in the production of the methane. In a meta-analysis, has been described that Asparagopsis, both of species can decrease on average 1/3 the methane emissions. And I have a PhD student which is evaluating the effects of Asparagopsis. And the thing that occurs when we feed animals with Asparagopsis is that there is a decrease in methane, that's fine, and an increase in the hydrogen. And we are aiming to develop strategies to redirect that hydrogen to products that could be used for the ruminant. In particular, we have supplemented animals with fluoroglucinol because during the degradation of the fluoroglucinol, the hydrogen is incorporated, and that could act as a hydrogen sink, leading to animals with lower methane emission and as well with higher productivity. And here, just to conclude, I present a meta-analysis in which the different methane inhibition strategies are evaluated. And here you can see that some of them have more consistent effects, for example, the lipids or the tannins, saponins, hydrogen sinks. For example, the vaccination doesn't work very much against methanogens. And then as well, we have to say that management can as well decrease methane emissions. And here I present some of I'm preparing a book chapter in which we describe the different nutritional strategies or mitigation strategies. Some of them can decrease methane production, for example, improve forage quality or increase feeding level, use of Asparagopsis, 3NOP, or lipids, or even saponins. All those can decrease methane production. Other strategies such as essential oils or tannins, they normally decrease methane yield because they increase productivity and ultimately they have a dilution of the methane emissions. So the take-home message from this talk should be that there are different natural feed additives available in the market. Some decrease methane production and others decrease methane intensity. However, most of them tend to have a high variability in their effects. So we should evaluate these strategies under the different production conditions in order to determine their efficacy and cost benefits, particularly if it's economically viable to implement those strategies. So thank you very much for your time, and I'm open to take your questions.
Alejandro, thank you so much for such an interesting overview of all the mechanisms and from plant extracts and how they can be used and how they operate. I can see Henrique already jumping on his seat. We talked about Asparagopsis, but let me just take a couple of minutes more from Alexander to pose some questions from the audience. I'm going straight to the questions of the audiences. And one of the questions is whether you have conducted any trials using Cistos ladanifer, which is estiva in Portuguese, extracts. So have you ever tried? Do you know what is the state of the art by using this essential oil?
Sorry, which essential oil you mentioned?
Citrus ladanifer.
No, the thing is the essential oils, there are hundreds of them. So now we are going to run a in vitro experiment in which we are going to evaluate in 20 different essential oils, but we didn't include that one. And that is important to do a very good screening of them, because sometimes most of the times the best is to combine different essential oils in order to maximize their effects.
Okay, I think this question also comes because this is a plant that's wild available here in Portugal, and you know, the only use for it is essential oils. And one of the things is try to understand whether they could also be, you know, redirected to feed. Alexander, we also have another question here: is whether is there a type of extraction that is better than the others? And I think you've touched that on your presentation. And when you extract, I don't know if you do extraction yourself, but which methodology you use?
I normally don't do the extraction myself because the different feed companies are experts on that. But yeah, that can have a very big impact in the activity, because during the extraction method, some active molecules can be inhibited, and then of course, the activity will be lower. So I will recommend to do the extraction with the less aggressive procedure possible, and then or use the pure additive freeze dry to try to minimize the inhibition of the activity.
Yes, and you also pointed that, you know, the costs of the extraction are also important to take into account if we really want this, let's call it, technologies to reach the market. Just one final question. This question was actually for Pekka. Pekka, you can type in if you want. But what are the forage quality parameters that predict methane production? Or is it just strictly dry matter intake, regardless of the quality? So we're talking about forage quality or methane emission.
Yeah, that's a good point. We have used in a couple of months ago, we published a paper about developing equations to determine to estimate the methane emissions in sheep. And we conclude that the main driver is the feed intake. And then the quality of the diet, it didn't appear very relevant, because the one thing is what we feed the animals, and the other thing is what the animal is really eating, because some of the animals are quite selective, particularly small ruminants. So we have seen that in cows, the fiber is positively correlated with methane emissions, but in sheep is not that much. We have seen that, for example, it's more relevant if we can take rumen samples and to analyze the propionate molar proportion, and it's giving you a better idea of what type of fermentation is occurring in the rumen, and that better correlate with methane emissions. It's negatively correlated in this case, that the composition of the diet itself. This is for sheep, but I have to say that for cattle, because they are less selective when they are eating, the fiber is a key element for estimating methane emissions.
Okay, great. Pekka, do you want to add something to this? You're muted, Pekka.
Yeah, it also depends how you express this methane. That is it total methane production per unit of per methane yield or methane intensity. That if, for example, if we have higher digestibility of forages, then it can increase total methane production. It can also increase methane yield, but certainly it improves methane intensity. So less methane is produced per unit of product. And of course it is also fermentation. If we have silages, fermentation quality can have some influence, and there are many different factors. But I think overall, we have made also some meta-analysis of predicting methane. As Alejandro mentioned, dry matter intake is clearly the most important factor. And let's say with the best model, if we can describe 100% of variation in methane emissions, dry matter intake certainly explains more than 90% of that. That diet composition has very small effect compared to total dry matter intake.
Okay, well, great. Thanks for that clarification. Alexander, just one final very short question. All the figures that you present and all the mechanisms, they relate both to milk production and meat production, correct? Or do you focus more?
Yes, the methanogenesis is common to dairy and beef animals, so it should work the same. The only thing is different is the diet, but most of the feed additives are working not exactly the same way, but very similar. For example, the 3-NOP is working better with intensive systems than with extensive ones. But so yeah, in general, we can apply the same principle for both systems.
Okay, thank you so much. Henrique, you're still there. Thank you so much. Now it's time for our super seaweed. Okay, thanks for hanging on, and apologies for running slightly late. So I now hand the floor to you. It's been such an interesting talks.
Thank you so much, Claudia. Thank you so much, and congratulations to the Food for Sustainability team. It's and for this academy. I love the concept, and it's really a pleasure to be here. I just want to make like a disclaimer here saying that I'm not a biochemical, so most of the of what was spoken here for me, it's a bit more difficult to understand. But I'm going to give my contribution and explain what's the role of seaExpert within this path of this research that it's been since 10 years now on Asparagopsis. So I'm going to share my screen as well. Let me see. So screen, right? I think this is the one. This is correct. And
I can see your presentation. Yes.
Yes. And now I'm going to put it into presentation mode. Right.
Yes. Perfect.
It's perfect. Okay. So I won't be very specific. I will not dare to mention all the chemical processes that are happening in the rumen. So I don't dare to go there, but I will tell a bit about what we've been doing in the last year. So the title for my presentation is Asparagopsis for methane reduction in cow feeds from the Azores to the world. So we are based in the Azores, in the Central Group in Faial, and seaExpert exists since 2004. But in the last 8 years, more or less, we've been dealing with seaweed. 10 years maybe we've been dealing with some seaweed. We've been getting some contacts and people asking for supply of seaweed for tests, for experiences, and including for Asparagopsis. So Asparagopsis in Azores, as Alejandro was saying, and Alejandro, I just loved your presentation. Thank you so much. And yes, I've learned that they also produce hydrogen. I didn't know that bromoform would make the cows to produce hydrogen more. I didn't know that. So yes, in the Azores we have both species, Armata and Taxiformis. They are both exotic, huge blooms of biomass available usually. It's used, this seaweed, both are used for several applications. and one of them is considered invasive, which is the Armata is considered invasive in Azores. And it's a species that goes down to 10 meters maximum, usual 10, 15 meters. And it happens now. This is the season. We are starting the season for Asparagopsis now. So here are some specifications of the seaweed. In the Azores of both, they are very abundant. Nowadays, I believe that Taxiformis took over again from Armata, but we can still find a lot of Armata available. Now competing with the new species, the new invasive species called Rugulopteryx, but that's another story. So the multiple uses of Asparagopsis, usually it was initially used as food. You know, Hawaii, the kings would have their own recipe for Asparagopsis. It was really appreciated. I don't know if someone ever tried Asparagopsis. It's not a very, for me, it's not very tasteful, but it has a strong flavor. But we also supply, but we don't supply usually for food. We supply mostly for cosmetics. So there is inclusively a Portuguese brand with using Armata from the Azores. And also as well now, as said by already Alejandro, it's for use for trials on animal feed. So we all know that livestock culture deals with a lot of problems. It has a huge impact with deforestation, water, waste. They step on the ground, so methane emissions. So there is a big social pressure for reducing impact from livestock. And gladly, back in 2010, 2011, someone, a livestock producer in Edward Island in Canada, he was walking with his cows at the beach, and he had this. He realized that every time that he would take the cows to the beach, they would eat seaweed, a lot of seaweed, and the ones that would eat seaweed, they would look happier and they would produce more and be more energetic and sociable, including. So he realized that there has to be related. It has to be related with the seaweed. So he went around and he spoke in some researchers in Australia, Rocky Denise and Dr. Rob Kinley. They decided to try a lot of seaweeds species, and in the meantime, so all were good, all were great, in fact, but there was one that had this like super, hyper, mega impact on the cows. And that was Asparagopsis. At the time, they realized that yes, Asparagopsis it might be the solution or part of the solution. Okay, so I will not explain this about the complex in between vitamin B12 and bromoform and the enzyme and so on. So you all know a lot about this, but this was the biggest discovery from. So since then, this is when it all started. In 2013, there is the first paper from Dr. Kinley, I think, saying that Asparagopsis was really powerful on methane reduction. And this is, like, then we have some points, important ones. And so back in 2000, in the late 2020, there was a paper released saying that if you put Asparagopsis in oil, in any vegetable oil, it can also be used, as the bromoform and other compounds are volatile and they go through to this oil. So it's a very good discovery, because it can turn it into a industrial product, but also brings some additional challenges. For example, for the US to do this oil, it becomes a drug. It becomes into the drug scope, and so it's different than to approve just the use of the seaweed. So now you use extract of oil of seaweed in oil. So it's harder to get to be approved. So in the meantime, since 2013, a lot of papers have been produced around the world on these themes. So I believe that most of you, you've heard about this and you've read and you participate probably in some of these papers. Alejandro, I don't know, Pekka. So now about what we do, about our role on this process. So back in 2013, we supplied Dr. Rocky Denise and Dr. Rob Kinley for their research. In 2013, we shipped dried seaweed at the time for their trials in Australia. In 2014, we did it again. So it was, at the time we got a bit surprised. In Australia, this seaweed comes from there, and they were getting seaweed from the Azores to them. So we dealt with a lot of dried seaweed, and then in the meantime, we turned in 2018. We tried in 19. We started with freeze dried. So that's when the papers came out mentioning that the seaweed would have to be freeze dried. So it's a very expensive process. It has a huge impact on the compounds of the seaweed, but was the safest one and the one that was mentioned by science. In the meantime, as I said, in 2020, late 2020, there was this paper that came out from Magnussen saying that you can drown the seaweed in oil and use the oil as a feed additive. So seaExpert has been involved in all of this. A lot of papers around the world have been published using seaExpert biomass, and we are in strict contact with all these research teams. We like to know and to have a proper feedback from what we do, because we make it a business, of course, but we care. We do care about what's going on, and we want to help on this research, or we want to know and supply more and more, because most of the papers were based on animals, on cows, and we want to know more about the seaweed itself and how to make it better, okay? And how to get the best out of each specimen and be useful, because it's a very very high responsibility when you harvest something from the wild. Okay, so we have to do it with a very strong sense of responsibility and the best available knowledge. And all these scientists can help us to be better and to improve the quality of what we supply. This is not a Walmart product, okay? So every year we are developing and trying to make it better. In the meantime, seaExpert did its own research and development on innovation. So a lot of papers have come out from using our seaweed, but then we also make our own technical data sheets on the Asparagopsis. Then we started this partnership with the freeze dryer company in Europe. And also seaExpert has been doing a research project with the veterinary faculty and the INIAV in mainland, in Portugal, with the production of oil. As you can see here in this picture, we are developing this for almost 2 years, 1.5 years, let's say. And now we have also another project that it's submitted. We don't know if it will be approved or not, with IPMA for how can we use the remaining seaweed after you put the seaweed in oil, and then you take the seaweed out of the oil, and you have to do something to find a way to do it and close the cycle of the circular economy and not just throw it away or burn it. So we are trying to find any possible application from it. So everything is most welcome. All opinions are welcome, and contributions. We had from previous presentations, we had already some good contacts of people saying, maybe we can do something. So seaExpert is really focused as well on finding a solution for the remaining biomass after we drown it in the oil. So the harsh reality in the world is that we have 1.5 billion cows. Let's say that, okay, in average 22 kilos of dry matter per day. If we give 0.2% of that food, it means of that daily feed, it's 66,000 tons of dry matter per day to feed all these cows. So let's say a big company, the biggest one in the world, in Australia, says that they're going to have 6,000 acres of production area. They say that maybe if they can produce 180 tons per acre, let's say they can feed 0.33% of total cows in the world. So this is a big challenge for the seaweed, and we have to really make the best use of it, of course. So that's why I say that we are part of the solution. We are not the whole solution, for sure. So it's a combination of everything that is available, as Alejandro and Pekka was just saying. So you can use several natural products to reduce. So all contributions are positive, we hope. So nowadays we have already some. So the future will be for sure production, and we have several platform supplies. I opted to expose this here to you all, for you to know. So we have wild harvesting is seaExpert and another company in the world that who are doing it. Of course, that it has some constraints, and it's not the solution. Will never be the solution like seaExpert. If we, our wildest dreams, if we could harvest 300 tons per year, we could feed 2020 cows per year in Azores, which would represent 1% of the cows in Azores. So, I think that wild harvesting is not the process, is not the solution, but we were the solution for research because no one is producing it yet. But, well, some works are really advanced, like marine aquaculture nowadays. Yes, they are doing a lot of research. Maybe it's the solution because that's where you have more area available, but the technology is not 100% improved yet. And also there is an environmental impact, some positive, some negative for Asparagopsis taxiformis, like the development is still, they are not there yet. There is a lot of research going on, but for Armata, yes, there is. But the production, it's mainly for now, it's for their own trials. For these companies that they are involved in partnerships with livestock producers. So they have their own projects, and for now their production is mostly to supply their trials. But still, we, like for example, seaExpert sold Asparagopsis in 2022 to Australia as well. You also have inland aquaculture, which will demand a huge area for production. Of course, inland or at sea, it both will occupy a lot of area. But usually, like coastal areas are really strongly disputed by all stakeholders, like tourism and for people to live in. So they have a lot of competitors. So it's a challenge to do it inland, at least in Europe for sure. Maybe in other continents, I'm sure that they have more room. Europe is the old continent where all the cars are small, and we need to find small solutions, never too big. And so finally, I would like to tell you the ways that this seaweed is presented nowadays, is available to the world for multiple uses, but now for, let's say, for methane reduction, for feed additives. So we have dried, which has it's dried in the dark. Maybe you can dry it in the dark, but there are some papers mentioning already, who tried already and tested the difference between dried and freeze-dried, and the freeze-dried is still better than the dried seaweed. So also the shelf life of this product is short. Then you have freeze-dried, where it's okay, it's a huge, but the biggest bottleneck of the freeze-dried solutions, I would say that are two. One, it has a huge footprint because it demands a lot of energy, and second, it's very expensive. Nowadays, it's very expensive. You could never supply an industry selling seaweed at 200 euros a kilo. Okay, so it's not an option. Even 10 times less than that, it might still be critical. So freeze-dried is not the solution, but is the solution for science, for research, and that's how we've been supplying so far. And then last year we already supplied some extract in oil. Everything is experimental, everything is new to everyone. So we are one of the few companies that are focused on the seaweed. So we don't focus so much in the cattle, of course, but we have livestock producers as partners. We have scientists on animal production as partners. So of course that we have to communicate and cooperate a lot. And it's with them that we learn. But our main focus is on the seaweed. And so I guess this is most of my presentation. I will just finish with the biggest challenges for the supply of Asparagopsis for the industry. So I think that the big challenge is to get everyone to cooperate and communicate. Most of this research nowadays for production of Asparagopsis are, of course, for private groups, but they are been funding with public fundings. So I believe my belief is that we all should cooperate because it can't wait. Solutions like this shouldn't wait too long and just being handled in the basis of competition and to see who gets there first. Sometimes
And
I
this
say
is
that.
why we are here, Henrique.
Yes, yeah,
And
that's it.
very
True,
different
Claudia.
backgrounds is to make
Yes,
sure
true.
that we all together
Yeah.
here to reach a solution. Henrique, that
Yes.
was a brilliant
Thank you so much. Let me just finish,
presentation, a very different.
Claudia.
Oh,
Just give me.
apologies.
No, it's okay. Just
Yes.
20 seconds, just more 20 seconds.
Good.
Of course, and then just to mention that it's the scale, of course, as well, is also a challenge, a big challenge for the future, because we have to produce a lot of seaweed, and research will have only its own questions. The more research we have, more questions we have. So as Alejandro and Pekka just showed us. And of course, to optimize seaweed potential. That's to make sure that we use it the best way and don't lose compounds in between harvesting and using it as a feed additive. So thank you so much. Thank you to all. It was a pleasure just to do this presentation. Thank you.
Yeah, thank you, Henrique. It was a pleasure to hear you also. Henrique, you have plenty of questions in the chat. I will, and also here in Slido, and just for the sake of time, which I don't control at all, as you can see, I just have a couple of questions for you. Then we'll go for the round question. But if you can then just have a look of the questions on the chat and just try to see if you can just provide us an answer. But one of the question is about the seasonality of the seaweed, if we can use it all year and all year round. And we also have another related question is whether, if we are going to produce this at a scale, will we have enough seaweed, and wouldn't we be causing another problem to using seaweed Asparagopsis as a way to solve methane emissions?
Okay, so yeah, thank you all for your questions, and I can reply to all, of course. How to address the seasonality? It's planning. You have to plan. So most of the projects, they are submitted and they have to make these calculations when they present the project, and when it's approved, they know that they will have to use this seaweed, so they have to plan and make orders in advance. This for wild harvesting, okay, because it's seasonal. You said that seaweed has a very strong flavor. Yes, cows are known to be very sensitive to the taste, and in some situations it caused some problems. So you have to reduce and give them the minimum with the maximum effects.
Henrique, it's just one question on that. Have you tested the effects on the flavor of the meat? Has it been?
Yes, there are some paper. We didn't. Okay, so we didn't. We supply research. So we do our own research, but on product development on the oil. But yes, they've been tested, and some are accused. Some they
flavor.
are sensitive. Yes. So.
Okay.
But not a problem when you use minimum doses, and that's where we are trying to get to. And how stable is the bromoform during storage? It is known. There are some tests being done, and yes, it lasts at least over 1 year. We know that we are considering that 2 years is a good shelf life for the product. What else? Yes, the bromoform is retained in Asparagopsis oil. About the impact, I always say that sustainability is about managing our hypocrisy. It's about managing our hypocrisy with knowledge, with the best available knowledge, and with a strong sense of responsibility. So there's nothing that we do that won't have an impact. Everything that we do has an impact. So we have always to balance things and to make sure that we are doing what we believe that it's the right way to do things. And then when you are weighting things, balancing, if you find that your impact is bigger than your negative impact is bigger than the positive impact, then you start questioning yourself and you may quit on this. So far, of course, that the growth of Asparagopsis can be a problem at sea, but there are many, many, many problems at sea. And if you grow a seaweed where you don't feed the seaweed, it just lives naturally by the nutrients available in water, where you don't add anything to the water but just some ropes and the seeds in the ropes, in a place where there already exists these species. Of course, if you are not introducing species in the natural environment, in wild environment, it might be
A possibility.
a possibility.
A
You
possibility.
might, yeah.
We need more projects. We need more research. We need to see how far we can go with these systems. Definitely, you are pointing towards a very correct way. So I'm going now to close this session. It's been great to have you here, but I would like just to ask you, the three of you, since the topic of this webinar is about how if we can really reduce methane emissions from ruminants. So what is your perspective? And given that the EU has pushed so hard for us to become a climate neutral continent, we had these quotas that puts the animal production in lower levels. There was the abolition, so we have again more animals. The methane emissions are already increasing. So I know you're doing a lot of research, a lot of effort, research and also commercial projects, a lot of effort to ensure that we're going to get there. But do you think we're getting there? Are we using the right policies? Is this the right way to become a climate neutral continents?
Well,
Alessandro,
I can only speak about Asparagopsis. So I think that my presentation was clear on that. It will be a big challenge. It's possible. The research going on already proved that it doesn't do any harm to milk or to production rates, to the quality, to the taste, and so from our side, I believe that these seaweed Asparagopsis can be part of the solution. Yeah.
Thank you, Henrique.
Yeah.
Alexandro.
Yeah, I will say that increasing productivity is key because like this we could produce the same amount of product with less animals. And as well, it's true that in recent years, many farmers are using feed additives. The problem is that the use of feed additives is not taken into account when we do the calculation of the inventories, national inventories, because we only take into account how many numbers of animals or what are the average diet, but we don't consider if they are eating additives and what is the effect of the additives. So in the future, we should do those corrections in order to have better figures. And another
and Alexandro,
area,
just
yes.
building on that, do you think we should also try to incentivize the circular economy to take advantage of the local natural resources to get these essential oils towards, more efficient animals?
Yeah, that's another important area because, we have different additives that it has been proven that they can decrease methane emissions. Some of them, they don't increase productivity. So the question is, who has to pay for that? Because we know that 3-NOP or seaweed or essential oils, they can decrease methane, but I don't know, the farmers shouldn't be the one paying for decreasing methane because they already are paying too much for the feed at the moment because they're increasing feed prices. So they probably the government should have to incentivize those strategies. I don't know, the European Union, or they could use that as a marketing strategy, and in that case, the consumers will pay the extra cost of having a greener economy.
Okay, great. That's good points also. Pekka, and your views. Is it really possible? You're muted, Pekka. We cannot hear you.
Yep. Yeah, I agree very much with Alejandro said about this. Who is paying the cost? That if there is not any benefit in production, then farmer or feeding cost or production cost will increase, and I think that is a big issue because they're quite seldom these additives, they have not shown any positive effects on production. That even if this amount of methane is decreased, it is still, if we think about total energy requirement of the animal, it can't produce very high production responses. That even if we decrease 25% of methane, then probably we can expect about 0.5 kilogram increase in milk yield if the energy partitioning is the same as we usually have, if the animals get more energy. But feed intake regulation is also so, as I mentioned, that if you increase energy from reduced methane, then it can actually lead to small decrease in feed intake. That we made some analysis of 3 NOP, and there was some small but significant decrease in feed intake, and that probably explains partly why we don't get any production responses. And I think that as Henrik said, that there is not any single solution there, that we need to look all these together, that small steps in different areas. And one thing I think it is very important is that we look the whole greenhouse gas emissions in production chains, and not simply methane. Like if we feed more concentrate, that can lead to some decrease in methane, not so much with the dairy cows. But if we take into account greenhouse gas emissions from producing grain or grass, then actually grassland-based systems can be more effective. And also feeding of human edible feeds to decrease methane, I don't think it is very sustainable system. That if we feed human edible oils or human edible grain to ruminants just to decrease methane, I think we will simple stomach animals use grain much more efficiently than ruminants, especially if we increase it from our quite high current levels in intensive production systems.
Okay, that's also a good point. So this is actually a combination of all the strategies, and that's what we've been talking about in this webinar series. We've talked about genetics, we talk about feed additives, we talk about management, we talked about the role of the animals in terms of turning the agricultural system more sustainable. We looked here about on natural feed sources. So I think it's a combination of all these techniques. I now come to call to a close this webinar series. I start by thanking these wonderful experts that we had today. I would also like to thank our audience for being here and staying with us for such a long time. If you want to revise any of the talks, we will have all this recorded. So thank you very much. We will start another webinar session, this time on functional nutrition. We're going to change the topic slightly. Of course, we're going to talk about plant extracts, surely, but this time for human food. So thank you again. It was a pleasure to be here to moderate this webinar. Thank you so much for your questions. And I hope you can join us later on other sessions. Okay, thank you very much. It was a real pleasure. Bye-bye.
Thank you.
Thank you.